Now, which is great. Good afternoon, everybody. Thanks for coming to this week's Raymond seminar. We have it's a very special guest, the department's very own Deborah. She really doesn't need an introduction. I mean, we all kind of know who she is, but it's a bit of a special occasion because she's retiring at the end of the semester. So kind of trying to highlight a long career here. This is kind of a bittersweet moment that she's been kind of like a central voice and obviously a big player in the department for a long time. So take too much time away from Deb, but I do want to talk briefly about some career highlights. She's been an active researcher for 35 or more years, which is crazy. That's longer than a lot of us have been alive. Not that yeah, so the experience is insane.
1984.
She's held faculty jobs at NC State, Toledo on the first year. And then when she first came here in 2004, she was actually hired as department chair, responsibility she held for 14 years.
Yep.
So despite the extra work being like chair, and that comes with a lot of extra responsibilities, she remained incredibly productive, publishing over 100 peer-reviewed articles in multiple different journals, giving some 300 talks, which again is insane. That's like a whole year of just talking about. And she was obviously was well funded, so I think her totals are somewhere $13 million. So continuous funding all throughout. That's a really impressive list of accomplishments, but she's obviously a lot more than that to us as colleagues, students of hers. We know her to be a great role model, mentor, and honestly just a good friend of the department. So she will be missed in retirement, but we're lucky to have her for an extra hour to kind of hear your reflection. So with that, I won't take any more time, and we can save all our questions to the end. Thank you, Deb.
Been a hard one to get ready for us. We'll talk about it. Where do we begin? So thank you for being here. Thank you for the very kind introduction. Before I get into the seminar, for those that came in late, I just want to say I'm cleaning out all my books and my files, and in there I have lots of reprints from the internet days of my publications that started in the 1980s through there's probably early 2000s. So please take them, otherwise they're headed for the recycling bin. I gotta clear out the office for the next one. Okay, so as you see on this quote here, this looks a lot like One Health, like we keep hearing about today. This is 100 years ago. It's not a new concept. We just keep putting new terms. And soil's long been recognized as a living entity, not just for scientists, but also artists, philosophers, and politicians. And here are some of the quotes that some of those types of people have made about soil. I mean, even Darwin talks about the life in soil. So we've known that, but what you're also going to see today is biology is way underrepresented in soil, monitoring programs. You'll recognize a lot of those names. I won't read them to you. So today I want to do 3 things. I'm going to give you highlights of my career path, and then I want to kind of give a history and where I fit into soil health. And then I'm going to talk a little bit about highlights of compost ecology research, which I've done here in Vermont. And then I'll just finish with some brief gratitudes. So here's the fun pictures. Not necessarily this one that's coming, but where did I grow up? I grew up right here in Northwest Campus. Population of my town was 900. That was the largest town in the county, right? So we were the big city. And we were happy we didn't have any stoplights or anything. So it was just stop signs. But this is a picture of the farm where I grew up. This is the pasture that eventually got planted to sunflowers. Kansas is the sunflower state, as you know. That's the barn that we had, and we had all up here, and these were some of the grain bins. We had livestock and small grains in the picture. So a little bit about my town. Kansas is famous for its sunsets, and that is no filters. That sunset, that's just from my house where I grew up. At night, we regularly saw the Milky Way because there's no lights. Amazing. But anyway, that part of the country was inland sea a long time ago. So we loved climbing these chalk bluffs and looking for fossils. A lot of shark's teeth, a lot of bones. People come out there for their archaeological digs. Very famous for that. We had the Smoky Hill, or the branch of the Smoky Hill Trail, so we had a reenactment covered wagon company in our town. So when you volunteer, they let you go on the trip, so that's neat. Wheat harvest is a huge thing, a huge cultural thing. You bring out the entire extended family for lunch every day. And my favorite was driving the wheat truck, but here we are having lunch. It's the only one I can have. You can see I don't have as curly hair then. And then I was really active in 4-H. I showed chickens and pigs and learned how to sew. And this is one of my things that I designed and sewed for myself. So that's one of my hobbies, all from 4-H. So my educational path, try to put some pictures. I went to a small liberal arts school in McPherson, Kansas, and can you see the X? It's right in the middle of the state. Okay. I was one of 3 students majoring in environmental science. It was the hardest major on campus. It was a combination. If you took environmental studies plus environmental science on this campus, it did it all. So we did, you know, it was a liberal arts school, so you had to. And it's gained some fame here recently in the last year. It's well suited now financially, but it's the only school that has an auto restoration program. And they just won second at the nationals for that restoration of the Mercedes-Benz. So when I was a junior, my advisor pulled me aside. He goes, "What do you want to do with your life?" I said, "I have no idea." He said, "How about research?" Yes, give it a try. So here I am doing research in undergrad, and I got to go to the Kansas Academy of Science with my project, which was super cool, me and the 3 science professors of the college. And I was 30 miles from the Land Institute. So that's where I first learned about it, was in the early 1980s, and it had a huge impact on my choice of career path and what my degrees are in. I spent a couple summers at University of Michigan's biological station. I had to show that a small college education was grad school worthy. They don't believe your grades when you're at a small school, so you got to show you can do it at a big school. And I also had a summer where I did more professional skills, kind of like they do the field naturalist program here, or like the Quest program, that type of thing. And it was super helpful in terms of kind of giving me the right footsteps. Okay. I'm the first natural scientist in my family. They're all social scientists and artists. So I had to kind of pave the way myself and ask lots of mentors. But I chose to go to University of Illinois to do my master's in plant biology. And they had a great theoretical ecology program. I got linked up with a tropical plant ecologist, and that was my first introduction to diseases in soil. I worked on a project that related to hers. It was so theoretical though, it was great, but you know, those things that are good for you, make you glad to move on. I went to UC Davis in plant pathology where I could get more of an applied thing. In plant biology, I learned all about plants, but when you go plant pathology, it's all about the pathogens. All they know are sick plants. And then after that, I did a very non-traditional postdoc at North Carolina State University. I joined up with a program called EMAP, and I'll tell you more about that. Did that for 3 years. And then I spent 3 years in a non-tenure track faculty position, and I taught graduate students on plant disease epidemiology. It took me 3 years to find a job. I think job markets are tough today. I interviewed over 13 times before I got an offer. My first offer was at Toledo. I was too agricultural for ecology positions. I was too eco for ag positions. Or I was the token female, or they shut off the search in the middle of my interview, or, you know, all sorts of fun things. But it was an excellent networking opportunity. I met people literally from all over the country, from California to Florida. And at Toledo, I was the very first soil ecologist Ohio had. Well, actually, I had one other, but he was doing more aquatic stuff. And now they have 3 or 4, so that's cool. Here's Toledo, and here is a summer camp that I'm hoping to lead. We're out at doing some remediation on heavy metal contaminated soil. And landed here, 2004. Okay, that's the background. And here's the history of soil science and soil health. And the little cheerleaders here are the steps that I was involved in during my career. Just to start you off, the discipline of soil science began in about 1900. Silent Springs in here, that's about the same time that people started recognizing the discipline of soil ecology. So many years later, explains kind of why the science is a little bit behind. We have a society named Soil Ecology Society. That's kind of where I started entering in the early 1990s. So I'll talk about these steps with the cheerleaders here in a little bit more detail. But my point is, soil health is not a new concept. It's been around for a long time. I got to be into it at a real active time in the early 1990s. I was so excited to see an entire issue about soil quality come out in 1992. It came out in the journal called American Journal of Alternative Agriculture. The journal later became Journal of Sustainable Agriculture, and today is Agroecology and Food Systems. That one, I think I missed a word in there. But I just showed you there's evolution of journal titles too. But this was a result of a workshop that was at Rodale Institute on soil quality and the initial thinking about we need some soil health, soil quality programs. The person who edited this issue was Garth Youngberg, and he was the director of an institute called the Henry A. Wallace Institute for Alternative Agriculture. I had the good fortune to serve on their board for a couple of years and then get invited back to help them with this special publication, which goal was, are we ready for a national soil quality policy? This is the table of contents. That's published in 1998. You can see the same table of contents probably today. So obviously the answer was we aren't ready. What all was going on in the early 1990s? There was a lot of activity going on in the Soil Science Society of America, and I had a chance to interact with some of those folks in various capacities. But it came out of that was a really important pair of documents on soil quality. They're cited here, but this is when they came out with the core set of here's what the functions of soil are. And you can, read them. I mean, it's physical, chemical environment. You've got, water movement, mechanical support, buffering capacity, source sink and filtering. And it was really clear, soil does all of these things. And if we want to have a soil quality program, we need metrics that measure those things. They came up with, after long heated debates, with this minimum data set. And a minimum data set means the idea is let's find indicators that everybody will measure and using the same method so that we can compare across studies. Well, that's kind of work, but kind of not. We're still working on that. But take a scan over here. There's no biology. The closest it comes to it is soil organic matter. We know only a portion of soil organic matter is living. But so that's kind of the beginning, and at the same time, kind of spun off some programs through the national or NRCS, Natural Resource Conservation Service. By the way, it just gained that name in the 1990s. It was called the Soil Conservation Service before that, and that started after the Dust Bowl in the 1930s. Okay, but they realized that was a little narrow. They needed to have a little broader perspective. And I think many of you are aware that they conduct these regular surveys. Some of those acronyms probably look familiar if you've taken Joseph Forrest's class, like the Sergo and the StatsGo and NATSGo. But when you look at that, there's no biology. And my dream was, Can we insert a biological indicator into that mix? I mean, it's already an entity out there, but I made some progress, but not entirely. They also developed a Soil Quality Institute and put out this little booklet called the Soil Biology Primer, and you can still find that online. It's a little outdated, but hey, you know, it's still a classic. I keep saying soil quality because this group called it soil quality. There was a huge heated debate. Is it soil quality or is it soil health? And their first stance was to call it soil quality. And there's a definition here. But, you know, it's the subtleties of different terminology. At the same time this was happening, this was my postdoc where I started with this program. We wanted to call it soil health. And I think we won because they eventually came around and started using the term soil health. But you know, health I think is a nice one because it's like our health. You know, health, you can measure it, it's quantitative. You can look at changes through time with some key metrics. So this program stands for Environmental Monitoring and Assessment Program, and it was designed primarily through the U.S. Environmental Protection Agency. I was a member of the farmlands, or the agricultural lands component. They had teams of other ecosystems, but our particular group, we had a specific cooperative agreement with Ag Research Service, NASS, and the NRCS. So here are some things that I gained from this experience. I learned a lot about politics among different agencies and how they don't like each other very well. I saw some really bad behavior at meetings, literally people yelling at each other. And I learned a lot about what not to do as a leader, because that was not very effective, right? But I think the people that are in charge of our agencies, it's kind of scary, but very insightful. You know, sometimes you learn best when you see something you don't want. I also got to work with some fantastic applied statisticians at North Carolina State University, and I still depend on that information today. So I really gained a lot of expertise in multivariate statistics, for example. And I got to go and take the NASS training and learn how to be an enumerator. So I got to go to their schools and ride along with the enumerators that do those little surveys, you know, that they publish every year. It was really cool. But that's just the learning tips. But I want to tell you a few highlights about the research that I did there. It's really a non-traditional postdoc. I did a lot of logistical support. I did a lot of administrative things, which are not what you aspire to postdoc. And I was like, I have to do research. I want to go into academia. So I tacked that on there too. And I took it on to my charge. One of my charges was to develop biological indicators. We outlined these 4 steps, and I'll just talk briefly about each of these. We had to first decide what are we going to measure. Then what index do we use? You know, how well does that perform when we're looking across space and time? And you have detected change, you know, with a reasonable sample size. What's a good site? What do you compare it to? You know, we're thinking you have organic ag or you have grasslands. I decided not forests if you're looking at ag lands. You really need ag references. We don't need to go back to pre-European times or anything. It's not very realistic. And then what does it mean? Okay, so what should we measure? I'm gonna have to speed up. It's like, okay. So we usually stick with what we know, because that's our comfort zone. I was a plant pathologist, I thought about plant pathogens. But when you're working across regions and all kinds of crops and ecosystems, that doesn't work. It's not going to be a universal indicator. So I had to get out of my comfort zone and start reading a lot of literature. Punchline is eventually came around to soil invertebrates, which I had never, and I still haven't taken a class in nematology or entomology in my career, even though that's what I spent my career doing. Okay, so here's the challenge in these programs. Kind of twofold. One is, well, first of all, we know biological indicators are underrepresented. Organic matter, microbial biomass was about the best we had. but it's really difficult to interpret. What does it mean? It's really noisy through time and space. And they're so broad, you don't know if the biomass is plant roots, fungi, bacteria, which ones, you don't know. And it can change values just within a day. Because bacteria have life cycles of an hour, a few hours. We go up the food chain into invertebrates. We have, and being an ecologist, this made a lot of sense to me. We can get up into primary, secondary, tertiary, and quaternary trophic groups. But the problem is we had a limited, incomplete taxonomy, and there are not very many people that have the ability to do that identification. But I was like, we have to do this. It makes sense. So here's some advantages of soil fauna over the microbes as indicators. Are you getting the right pictures? The entire the food chain, as I said, and I illustrate that the food web over here. And so I would argue they really integrate all the information that they offer plus everything below it. So not only them, but the microbes, the soil chemistry, and the physical properties. So that's, and then generation time, I mentioned that relates to the variability. Microbes reproduce every few hours under ideal conditions. Invertebrates can be days to years for life cycle. So it's a little more stable. And most importantly, it tells us something different than the other indicators. I had to be challenged by all these NRCS soil chemists and say, why don't you just measure soil carbon? That'll do it. And I was like, it's telling something more. So I was convinced that I was this young little squirt that everybody thought I was an undergraduate when I was on the faculty. And I said, so I did it, and lo and behold, I found soil properties and microbial biomass predict less than a quarter, less than 25% of the variability associated with soil invertebrates. So it's telling us something different. So that said, I don't think you want to do it by itself. You need to build a nice maybe three-legged stool, things that tell you different things, and then collectively you get a more comprehensive picture. Oh, I forgot this one. And also, including invertebrates is really important in interpretation. This is just a simple diagram. I'll talk to microbial ecologists, and they say, who's responsible for decomp? Microbes. But sometimes the data don't work out right. So you really need to look up a trophic group level or not. Why did they go down or up? And it could just be because I got all eaten up by the predator above them. So just simple data. We're looking at decomp here. Whole system here, we have decomposition, percent weight loss, bacteria, and then bacterial feeding nematodes, and then mites that eat nematodes. If we add an insecticide, we knock out the mites, things change. We lost the top predator, therefore we have more nematodes. With more nematodes, they're going to eat more bacteria. If you have fewer bacteria, you're going to have less decomposition. So when you're interpreting, why do I have less decomposition? It's not so simple. And it's more informative if we look at least another trophic level. That's one of my favorite examples. So I landed on nematode indicators. I won't get into long time discussion, but I did a lot of statistical things like looking at estimating variance at different spatial scales, looking at statistical power, how many samples do I have to take to detect how much change, all the things you have to think about when you're looking at a monitoring program, because the whole idea is to measure change through time. I landed on nematodes, and long story short is I landed on an index that looked at ecological succession. Why nematodes? They represent at least 5 different trophic groups right in the center of the food web. You have your plant parasitic nematodes. Ecologists would call it herbivores. Plant pathologists call them parasites. There is a difference, and then you have those nematodes that feed on bacteria or fungi, and then you can have omnivores and predators. So you've got, these different groups. Okay, and so the other thing you might not know is nematodes excrete ammonium as a byproduct. They get extra nitrogen, they excrete ammonium, and that's a form that plants can take up. But they also indirectly contribute to nitrogen mineralization by grazing on the microbes and releasing nutrients that are bound up in their bodies. And also the nutrients that are bound up in their living bodies called immobilization. Try to make this, I think most of you know what ecological succession is. I'll really oversimplify it. But, basically you can look at when you have a heavy disturbance, you set everything back to, pretty much bare ground. And if you let things go, it'll naturally, progress into a more complex system. You can do this with nematodes too. If you look at, they have different life history characteristics. These guys are really sensitive to tolerance. They have really short generation times, right? These guys are really sensitive to tolerance. They have longer generation times. I mean, those are just simple, but it ends up, we tend to see bacteriivores and plant parasites here, the fungal feeders come in here, and then it takes out here until we start to see the omnivores and predators. But we can look at our whole sample, compute a weighted mean, and we come up with an index, and that person who designed it called it a maturity index. As an ecologist, I would say, what's that mean? It's really succession. But in the literature, it's called the maturity index. Okay, so I'm just going to get some highlights. So one of the big questions was, what's a good reference site? And I mentioned organic farming, pastures. Through a series of those studies, basically, I came to the conclusion that it's the physical disturbance and that tillage that really disrupts the soil food web. And so something that's untilled, like a perennial grassland or pasture, and we came up with 10 years or longer, not tilled would be our reference. So you can see a few key references, but I keep testing this hypothesis in my soil ecology class, and it keeps holding true. We keep seeing more and more data that support its soil. So now that's why there's so much emphasis on reducing tillage, Joseph, recognize that picture? That's what we did on a collaborative project. It was a slice of soil with the aggregate outline. So why is tillage hard? This is another way you can visualize it. Work I didn't do, but soil food web without cultivation, this kind of land. Add cultivation. Look at all those groups you just knocked out because you're knocking out their habitat. You're changing that soil fabric. It's the omnivores and predators that go. The fungi start to go down. These kind of soils tend to be bacterially dominated. Okay, so we get on to interpretation. Policymakers could care less what a nematode looks like. Your nematologists think they're adorable, but not a politician. They're like, why the heck do you do that? You could have picked a koala bear or something, something a little more charismatic. But what they do care about is that their lands are working, and that they're functioning. So I have always said we have to have indicators that reflect ecological process. And the two big ones in soil are decomposition and nutrient cycling. Early studies showed we had a nice, strong relationship here between nitrogen availability and our index. So, less disturbance high up, more disturbance down here. Disturbed soils tend to be the ones that's our ag soils, high in nitrogen, ammonium and nitrate. Once we got into less, like forests and wetlands, we could refine that, look at lower concentrations. And we were able to actually quantify the nematodes were contributing up to 22% of the nitrogen mineralization. Another comment I could make to microbial ecologists. It's more than just microbes. You got to look a little higher in the food web. Now, nematodes and nitrogen mineralization are pretty well connected, but if you look at decomp, the one that explains the most are really the microarthropods. And we estimated that was, up to 43%. And these are some of the groups that we looked at. I just put North Carolina because that was a study that we did there. It was one of my first grants that we had. It was one I totally under budgeted for. So I totally was, to do a better job the next time. But we had 3 ecoregions. We had geography there. We had wetlands, ag, and forest, and then pairs of relatively disturbed and undisturbed in each of those. And sampled 18 times over a period of 2 years. So we could get all the different seasons and also figure out what time of year is the best time to sample. We could do ecosystem comparisons. We did learn that you have to interpret these by ecosystem. A number of 3 for a wetland means something different than a 3 in an ag system, for example. So the next step was to go to the larger scale. For doing national monitoring, you need to start by statewide studies. I was living in North Carolina at the time, but we also went out to Nebraska. This is where I learned how to be an enumerator with NAS because we worked with their sampling design. And these are the sites that we sampled. It's color coded by the values we got for the nematode communities. So I went to all their schools. I got to train everybody on how to take the same kind of little cores and packaging them up and sending them to us. It was quite, I became the logistical person who decided that was enough of that. That's a good experience. But then we were able to then present these as what cumulative distribution functions. The y-axis is land area. So we know what fraction of the land area has what kind of index value. I won't go into details, but I think it's a method that we want to explore here. And then we could also look at the range of variables we get of these indices values across the states, and also kind of the distribution of the different indices, by state. Unfortunately, the funding got cut off after about 6 years. So this is about as far as we got. That seems to be the way government programs go. They last about 6 to 8 years, and then you change administrations in Washington, and all of a sudden they have a different interest. So they stop that one or something else. Okay, so where we're at, we selected the indicators, we evaluated them for performance, we made headway of calibration and interpretation, thinking about scale, ecosystem specificity, and that they are quantitatively reflecting ecosystem function. Now, this is where we're at today. We have a lot of work to do on calibration and interpretation, and even moreover, to have access to non-specialists, because some of these methods have a pretty big learning curve. But we got to do the fundamental research before we can make it simpler, so we know which taxa to put into our molecular kit. And this is the symbol for the Soil Health Institute. And I think this is an area they're really contributing to, is trying to link with farmers and the layperson and, bring that into their world rather than just an academic world. After I did EMAP, I had the honor of joining this group for 8 years. It was just a service thing I did on top of being a professor. But I got invited because of my experience with EMAP. State of the Nation's Ecosystem. If anybody knows Al Gore, this was one of his ideas. And so they funded this project with the idea they put out every 4 years, the State of the Nation's Ecosystems. All these different ecosystems were represented. I was on the farmlands group. One of the things I loved about this program, they were nonpartisan. Instead of asking, what can we do or what are we doing? They said, what should we do? And they had the guts to the blank graphs in their report. The idea was hoping to leverage some legislative funding to support the research to help fill in the gaps. Unfortunately, that didn't happen, but they did lay the framework, and it is out there. It's archived. I've got copies there. It's a little hard to find, but they really worked hard to make sure it didn't get lost. But, this is the proudest moment of my career, was that they chose nematode community indicators as one of the 10 or 15 because of my research on all of that statistical reliability and calibration and references and all that. So I got this really beautiful plaque. Thank you, provide. They don't pay you. This is all free service, but I do value that plaque a lot. I'm not going to talk about everything I did in my career. These are just other topics that I did. This is my transition slide to compost. But I've done some work with ecotoxicology when I lived in Toledo, looking at got me into molecular techniques, doing extra long PCR and DNA adducts, biotech risk assessment, some work with climate change that was funded by Department of Energy. And then we got to do some cool stuff in Costa Rica trying to quantify biodiversity. So On to compost, I'm just gonna hit some real highlights. It's gonna be slightly disjointed, but compost is not just a heap of decaying material. It has a very precise definition and a prescribed process and prescribed recipe that you need to follow. If you do it right, you get this beautiful like heat generating pile. This is a very complex graph. I'll just point out, there's a lot of changes that happen through time. I've already talked about ecological succession. Well, I love temporal patterns. And I can see that I can apply the temporal pattern idea again in compost and look at ecological succession. So that's how I justify getting into this field. Plus, I was in Vermont and love compost. So just looking at temperature by itself, when we get everything mixed properly, it heats up really fast. This is due to the respiration. And then, they start to run out of resources, oxygen, for example, and it dips. And so you turn it to keep it hot. The regulations say you have to do this thermophilic phase here to basically kill your pathogens. It's developed on human pathogens. It also works for plant pathogens and weed seeds. But what I was really interested in is this part. All you have to do legally to sell product is go this far. But you're losing some ecology because this is where all the beneficials and the biocontrols recolonize naturally. And so I got really interested in, can we look at, so they've got microbiome idea. And we have those, competing or, with the pathogens and kind of keep disease in check. Or another word is disease suppression. So, okay, so I can't go into details, but basically no compost is alike. We did surveys of all the commercial composters in the state. And they're all different. We found that what kind of carbon, they just tell you what to do a carbon to nitrogen ratio, but we found which kind of carbon you do makes a huge difference. You can see different kinds of carbon here, and even how you cure it, what process you do makes a huge difference. These all started at the same point, but we cured them different ways, and they turned out completely different. So it's not just throwing things together. I mean, you follow some precision when you make that cake. You don't just throw everything in your refrigerator in there and hope it works. This is complicated, but it's the idea. We did look at quantifying the bacteria and fungal communities through time, so we could see how they were changing through these different times, and you can see different colors mean they go up or down. And we also played around with, this is an undergraduate honors thesis that got involved with this, where we looked at it as we assemble different plant soil systems, how that changed the community in the soil. So we had basically unamended soil is here, and this is phylum level, and this is family level. You don't have to know it all. You can just look for color differences, okay? And then here's when we added vermicompost to the soil, how much it changed it. Here we added radish to it. Then we added the pathogen. And we split it up spatially. So we looked at inside the root and then the soil right around the root. But look at the cool patterns that happen here. And we're all like, trying to look for indicators, ways, that you can measure that. But unfortunately, this is the pathogen that we've introduced and it showed up in the root, so we know it was working. Just quickly, another big take-home message is we do know that compost can be suppressive of soil. This is a plant assay with radishes that we developed. The horizontal, the axis is kind of reference minus, without the pathogen, and then with the pathogen is without the pathogen. When you add the pathogen, it ended up, we got more disease when we had poultry manure in the system and less disease if we had dairy manure or hardwood bark or hardwood. And that fits pretty much what other people are finding, so it's not really exceptional. We went out to the field. This is Spear and Swift Street. We had a couple experiments. We just, this was with Food Safety and Modernization Act with the FDA and USDA. And this is a survivorship curve. We had raw dairy manure and, well, actually, yeah, that was something different. We sprayed E. coli at known amounts on the field and we could track its survival with different types of compost recipes. Everything, E. coli dies off pretty fast, except for when we had poultry litter in there. We're like, oh no, this is what all the farmers here are using. It's cheap. So we did another study trying to characterize what's different between poultry and dairy. And we narrowed it down to a couple families where we could see fourfold more in the poultry than the dairy. And you can get into the ecological interpretation, but it has to do with E. coli and a lot of pathogens really like nutrient-rich conditions. Okay, a lot of the other biocontrols tend to be better in less rich in conditions. We're able to culminate in, I was invited to write a chapter for this book. Survival of compost is about that thick. Some of you take my composting class know that. And one of my heroes, Harry Hoytig, who was known for the disease suppressive work in 1980s. And it was a real pleasure for me to get to work with him and learn from him. And we also had fun developing a video on some of our techniques, our lab techniques. And I kind of cheated. I downloaded it into my streaming site, so you don't have to have a subscription. And during the pandemic, I worked a lot with the Compost Association of Vermont. We did a lot of webinar series on soil and water quality and compost. Here's my gratitude slide. One of my favorite things to do is travel. I had a chance to present talks and do research in all of these countries during my career, which was, to me, just amazing. I feel proud of these. A lot of it's service that I did, plaques, especially this one. They named an award after me for starting the soil ecology section in the Ecological Society of America. So Deborah and Neher got the Deborah and Neher career. My brothers have made it. And I just have to say, and I'll embarrass Tom, I couldn't have done everything I've done without Tom White, who's in the back, being hiding in the lab, doing all this. So I just really, really grateful. I've had a partner. We met 38 years ago and learned we worked well together, and it's been our family business. So he's had a ton with working with my students in the lab. He's the walking encyclopedia. I think I'll stop there. So, Forest Ocean, you can ask me anything. Doesn't even have to be about science. It could be just about career. Yes.
Just all the countries you traveled to, I'm just curious, what was your favorite?
Oh, gosh.
Favorites.
Cool. I especially was happy to go to Cuba. That seemed really, really special. It was a very unique experience. I felt like I was one of the few people that were able to do that. And I was able to go back last May to, that one was really special. And I liked them all. I got to live in Wales for a year for a sabbatical, which was really fun. I'm this on organization of nematologists of Tropical America. They're the ones that have meetings in all the cool places, like, from Portugal to all the South American countries here. Those are amazing. I got to go to Machu Picchu, that was of course cool.
Egypt was also on.
Egypt was amazing, and we finished that this past fall, like a week before the war broke out. So that was interesting. We didn't have any sense that was coming. South Africa is amazing too. So much diversity. Oh, in Ecuador, I got to stand on the equator. That was pretty cool. We could go on, but those are just some highlights.
How healthy is the soil on the equator?
I fit on concrete. It's funny because one of my colleagues in Costa Rica thinks it's funny when the sun doesn't rise at 6 and go down at 6. Because he lives near the equator. And he comes and he goes, I am so confused. I don't know what it is. It's changing all over the place here. Here and,
I think Bethany's hand was up.
okay.
I love advice, both, I'd like some advice in the academic sense as like a new person in academia.
Okay.
And then, so more of like the academia side of a piece of advice for like a career path in academia. And then, more on like the personal side of like maintaining balance, maintaining your.
I anticipated your question.
Here we go. I'm scared.
Well,
Be
this
prepared.
is the reflective part, you know, and this comes over, these are some things, I think it's really important to stay curious. That's all part of learning. If you're not curious and inquisitive, you might as well throw in the towel. Focus, one of my master's advisors, I had 2 of them, said, focus on the science and doing quality science. Don't ignore all the stuff going on around you about all the inequalities that are happening or whether you feel like, you know, you're not being treated. Just focus on the quality science and it'll work out. That was it. Get ready for failure. It happens all the time. You fail, I figured 10 times out of, you know, for every one success. That's just the way that tells you you're learning. You're being original. No one's done it before. So you have to play around and try it and not be afraid of failure. That's just part of the process. Trust your intuition. I learned that the hard way. I went into one of those programs. I had my little proposal. I had an advisor that didn't like what I did. He wanted me to switch. It was a disaster. I switched advisors, went and did my project, and it worked out great. So don't, you know, just because they are older or have more experience doesn't mean they know your system better than you do. Right? Success is the best revenge. This happened, I learned that when I was just about ready to throw in the towel, and then one of my colleagues said, you know, so it motivated me to be successful and keep focusing on success. I think it's important to have healthy outlets for stress. Physical activity is one. I mean, music is something else other people do sometimes. Learn to laugh, be humble, kind and respectful. We live in a small group. You might as well be nice to people. If you start grumbling, then it's going to come back around to you, right? Everybody knows everybody else in your field. And that just makes life more enjoyable. And organization and time management have to be your plan that works for you, not somebody else. Because you can't adopt what someone else does. Our brains are all wired differently. So those are a few of my advices. Yeah, and Scott? Or who was it?
It was a very similar question, but I have a one that emerged
You
from
wanna
that.
add?
Yeah, well, what was one of your best failures?
Oh, what was one of my best? I mean, I've had a lot of grant proposals that never got funded or saw the light of the day. I've had cases where I really had falling out with colleagues that didn't work out. Those are all learning lessons. One of the things is when you find people that you work well with, become their golden. Because those are the rare ones. You know, you can, so, let's see, other failure.
Well, do you have a failure that like, you're in
Really, I'll get home and I'll think of it.
Yeah,
Yeah.
in retrospect, like, yeah, I'm so glad that didn't work because.
Well, oh, I was really into pursuing the ecotoxicology work and trying to come up with molecular indicators and tools for looking at sentinel taxa to simplify these nematode indicators. That's where I was headed in Toledo, and then I got this job, and then my whole life switched. Every time you switch, you have to change projects. Because what you can get funded there, and I could, you know, so.
Toledo had a lot of Superfund sites.
But
Toledo had a lot of Superfund
yes.
sites. That was part of the motivation.
Toledo is not a land grant school, so, you know, you don't get any land grant support there at all, and so how am I gonna test indicators? But a lot of industry, you know, that's like the auto industry field, and a lot of fields, you know, they call it brown fields, so contaminated with organics, contaminated with metals, mixtures. That was quite an experience. I went into a site. I had to talk to the EPA, and I said, "You're on your own. Don't blame us if you get, you know, harm your health or whatever." Try to PAHs. We had it so thick that you put your core there, and you go boing. It was like rubber, and you would have a pouring rain outside, and it was dry as bone. That stuff is hydrophobic. That is creepy. I know I had other failures. I hadn't focused on that. I was trying to focus on the successes. But the other thing is, we don't want to, I really have tried hard not to have regrets, but try to look at them as lessons. Because it's the glass half full or empty. You know, we can look at it one or the other, but it's a choice. It's not always easy. I'm not saying it's easy. I've certainly gone through my turmoils. Yeah.
I was fascinated by one of your first slides, and I think I recognized Diana Wall. Is that possible?
Yes, it is.
So I used to work in that department, and I remember her very well, and how much, and she really pushed to see soil as an ecosystem, yeah, rather than just a substrate. And so I was wondering, in your experience, how did that change in the approach and the mentality, and what kind of frameworks did you have to create just for the soil, just to see it as an ecosystem? Because I feel that in ecology, we had ecosystem modeling and approaches and frameworks, but I wonder whether they can be applied as easily for soil.
I'm so glad you mentioned her because she's really been pivotal in the field. And I don't know if you know, she passed away
Just a few weeks ago. Yeah.
We just lost her. It's really, really sad. I mean, she had just turned 80, but she had the energy of a 50-year-old. It was phenomenal. And she was a real pioneer for women in science. She was really pivotal. This is her book that she edited, which was Ecology of Nematodes. It was her work in this book that opened my eyes to nematode communities as indicators. And I had a really important phone call with her, and she convinced me that that was a good way to go. So she was always a champion. I think one of her massive contributions she has is to bring soil global biodiversity. I mean, bringing soil biodiversity to the attention at a global scale and trying to get policymakers to embrace it and fund programs that value it. So she started a program called Global Soil and Biodiversity Initiative. She's been very involved at FAO and all that. I mean, anything. And she also spent a whole career in Antarctica with that research. So, I mean, that's why I put her and these guys here as really, these are the founders of soil ecology, I think, in the US. So I'm so glad you mentioned her. So I just didn't, there was too much to go over in this slide. But good eye. Any others?
Favorite nematode?
I know my name. Tom knows it by appearance. Let's see. Was it a crowley? Which is the one that has a little crown on its head?
Crowleys.
And then another one. So soil health isn't like new as a concept, but it feels like industry is kind of going away from soil health, at least like repeatedly almost.
Yeah. You mean
What are
like
your
what
thoughts
kind of
on the
industry,
dry-
like chemical industry?
Yeah, and like just farming in general. Agriculture in general. What are your thoughts on the drivers and like that?
I mean, that's a tough one. I mean, how do you get people to change? That's a whole issue. And they're struggling, farmers struggle so much with the economic pressures that they can't always even do what they want to do just because the economics aren't there for it. That's one thing I learned a lot from growing up on that. It's just a lot of heartache when you can't do it that way. I do think Soil Health Institute, this is where they're really pushing it forward and really connecting with farmers and doing on-farm trials and trying to integrate some of those businesses and groups. I mean, the purest in soil. I'm like, oh my gosh, they're doing that. But, you know, I think it's a really needed area and I really give them credit for moving the needle on that. And thinking nationally in that effort. They've done some really cool, like North America long-term research sites and collecting some data. You know, we just reviewed that with the Shrek team. And they were recommending some of these indicators. And I would say the methods, they're testing some of these methods and minimum data set that I think will coalesce on. And they came after Cornell. The cash test was there in about 2005. So, you know, they've been doing a lot on that too, but I don't think we want to depend on just one university. So that's one of the things we're trying to correct here because we want to serve our farmers and the nuances there and add. I mean, you can only do so much, so. And I guess one other advice on just career-wise that I always tell my undergrads in their class was focus on what my next step is. Don't worry about 10 steps ahead. Because if you go through there, and this is my experience, you don't even know what is in there once you get the door. I'd never heard of soil ecology before until after I finished my PhD. And I walked through the door and I started, oh, that's cool. Yeah, it's just, you take one step and then you realize there's all these other doors you didn't know were there. You choose one more, walk through that, and now you have another set. You just don't know what you're going to find. And if you don't have it so fixed, then you can adapt. I think another thing is to think about adapting. Life change happens. It's all the time. It's inevitable. So be prepared. Times are going to change. You know, funding strategies change, administrative change. You're going to change jobs. You're going to move to other places. And we have to have, you know, be adaptable too. I think that's where a good solid education is really valuable. So that it allows you to be adaptable.
No questions from Chad either, so let's thank them one more time.
Thank you.
Thank you. Help yourself to refreshments.